Okay, so welcome to the webinar for of the Finnish Society for Bioinformatics. I think today we are going to have the talk from Mr. Dewei Hu, and who's actually now a PhD student at the University of Copenhagen. I think he has been working on this string project for a very long time, and I know that string is probably very widely used in the community by a lot of people to search for protein-protein interactions, and it's very comprehensive and has been widely used by the community. And today he's going to show us how to combine the string database with the AI for some analysis. Okay, so I think the floor is yours, and please start the way.
Okay, thank you for the invitation. And so I'm Dewei. I'm from the Novo Nordisk Foundation Center for Basic and Metabolic Research. And before that, I was working also as a PhD student at the Center for Protein Research at the University of Copenhagen. And let's talk about the strain database first. And if you're not familiar with the protein-protein interaction networks, I will give you a very brief introduction. So here we have a network of proteins, or you can call them the genes if you want. And if two genes are connected, it means they have maybe have similar functions, or they have physical interactions, or they in the same pathways. And we collect this kind of information in the string database from different sources, like the genomic context. For example, if two genes, they are the neighbors in the genome. And also we have the high throughput lab experiments, which are not done by us, but we just collect this information. And also we use the single cell data to do the co-expression of the genes or proteins and collect a huge amount of data by doing text mining, which is most important in string database. And of course, we just make use of the previous knowledge in other public databases such as KX or BioGrid. And we cover a lot of the proteins and from eukaryotes or non-eukaryotes. But mostly important, today I will just talk about how we do the machine learning on the eukaryotic species. And it's not very easy to, or not very straightforward to do machine learning with the graph or networks such as string. When you have a graph, you can probably do something like the unsupervised learning. You generate the embeddings, and then the embeddings could be features to represent each protein, and you collect some other labels which you want to predict. And then we typically can use this node-to-vec method to generate the embeddings. And this method starts from the random sampling, like for each of the proteins in the network. We start and then we take a random walk from this node to this node to this node, and we generate something like text or sentence. And once we have this tons of the protein sentences, we can use a language model. Here we can use a model called skipgram, and it can generate the embeddings based on the contextual information in our sentence, or basically from the information in the graph. So for example, here there are two clusters, and the clusters blue have very close embeddings in the latent space. And the cluster yellow or orange, they have very similar embeddings. And they can represent the pathway information or functional associations. It is quite useful to use this kind of protein embeddings in a lot of tasks. For example, during this summer, I spent 3 months with a biomedical consultant or a company called DDS, and we did a project to map the proteins to disease or do some disease protein prioritization using the network embeddings. And here, typically, we just start with the node-to-vec embeddings, and we have the omics data, which are used to define the proteins which are associated or not associated with the disease. And here you can see from the black line, which is our embedding-based method, and it's so much better than the traditional omics-based analysis. This is good, but there are a lot of limitations. One of it is it can give you comparable information across the different species. For example, we have the ACTB gene or protein in human and also in mouse. And since we are very close in the phylogenetic tree, and we have very similar protein function in these two proteins, and even they are very similar by the sequence. However, if you just use the node-to-vec, you cannot get very meaningful embeddings that, allow them comparable in two different species, because the networks are separated. And also you cannot train the node-to-vec on two separated networks. So what we are thinking about is that we can create something cross-species, because many of the string users, they do some research on model organisms, and they want to generalize this kind of knowledge. And let's say you find some target protein in mouse, and you want to know if it also works in human. So we want to make this cross-species embeddings for proteins in networks. And also, as we are holding a very big database, we want to provide this kind of resource to the community, so everybody don't have to recalculate every time. And how we are going to do it, we do it through the way called embedding alignment. And as you know, we have a lot of proteins that are evolved from the same ancestor, like we call them the orthologs. Let's say if there are two proteins, ACTB from human and mouse, they evolved from the common ancestor, and even after many speciations, they still have a lot of common functions, and they share some sequence similarities. And also we have the collaboration with another group from Madrid, and they do the database called EGNOG, which covers almost all the species from string. So you can find the ortholog proteins for any species in string from this database. And the embedding alignment is the way that we make the embeddings from different distributions as similar as possible. So for example, let's say we have two protein clusters from human and mouse, and from node-to-vec, their embeddings are completely different. But according to the orthology, they share a lot of the similar functions. So we should try to push them as close as possible. And so theoretically, after embedding alignment, they should be in the same cluster. And this is the same for other protein clusters. And we do this with a method called FedCoder. So firstly, we just generate the node-to-vec embeddings for each species. And it's very simple. We just run the code from public code repository. And then we just re-implemented the FedCoder, but we make sure that it's the way that we want. So we have the autoencoders for each species. And as you know, the encoder would project the original input into a latent space, and the decoder would reconstruct the input from the latent space. At the same time, we try to reduce the distance of those orthologs that I talked before, and to make them as similar as possible. However, because in our string database, we have more than 1,000 eukaryotic species, and it's not very ideal that we put them all together. and because we don't have so much GPU power, and also we actually have a lot of the species which are not very well studied. So if you put those poorly studied species with those well-studied model organisms, you will probably destroy the original information in those well-studied organisms. So the way we came up with is that firstly we do the alignment of some species that are well-studied. So we call them the seed species, which were selected from each branch of the phylogenetic tree. So for example, we have some animals, we have fungi, plants, and also some other prokaryotes. And we selected those 48 species and do the embedding alignment at the way as the same as I described before. Then after this step, we fix the embeddings of those 48 species, and we try to align all the other single non-seed species one by one. So once we have a non-seed species, we also have one autoencoder, and in the latent space, we fix embedding of the 48 species and try to reduce. We only change the embeddings of the non-seed species and try to reduce the distance of the orthologs from the non-seed species to the seed species. And once we get these embeddings, we want to make sure, like visually, they are aligned. So we selected 5 species: human, yeast, Arabidopsis, and this is slime mold and rat. And actually, rat in our definition, it's a non-seed species. And let's first look at human and rat because we know they are very close in the phylogenetic tree. So they definitely share a lot of the similarities in the UMAP realization. And if we compare the yeast and slime mold, and as you know, this is yeast, this is fungi, and this is not, but still they share a lot of the similarities. And definitely there are some of the proteins that are unique in yeast. And overall, if you compare the plants with any other species, you can notice that there are a lot of the proteins that are unique in plants. But if you compare the animals with the 3 species, you can notice that we also have a lot of the animal unique proteins. So this makes sure that we align embeddings as good as we want. And then we did a quality control with the CAG pathways. So in this setting, we tested a lot of the species, but here we just show 12 species. And in this benchmark, or more precisely, we call it the quality control. And so we are checking if 2 given proteins are in the same pathway or not. And in this benchmark, we just use the cosine similarity of the embeddings. So in each species, we have 3 types of the embeddings: the aligned embeddings, the original low-to-back embeddings, and this is the sequence embeddings generated by the Proto5 language model. And firstly, let's compare the network embeddings with the sequence embeddings. As you know, the sequence information cannot capture any kind of the pathway information. So definitely in all species, the network embeddings are always better or so much better than the sequence embeddings. And let's check the aligned embeddings and the node-to-back embeddings. And in most species, they just perform very closely. But in yeast, it dropped a little bit. And after we check the whole string database, we found that in most of the fungus species, the aligned embeddings dropped a little bit after we do the alignment. But we never know what's happening during these alignments. But we speculate that because we have the best quality network for yeast, because it's the most well-studied model organism. So we speculate that because we cannot introduce the new knowledge when we're doing alignment. So we would definitely drop a little bit from the best quality of the network. And then we did two downstream tasks to check whether our embeddings performed good or not, and also if you put the network-based embeddings together with the Pro-defined sequence embeddings, if it can improve the prediction or not. And as I talked before, the Pro-defined language model can give the sequence information give you it can give you a lot of information, but not the information from the pathways. But our aligning embeddings can give you the information from the network. So if you just very simply concatenate them together, you will have both the sequence information and the pathway information. So in this task, we call it the subcellular localization. We're predicting whether whether a given protein is located in a single localization or not. And here we have 10 different localizations. And we are using the data set from a transformer model called DeepLog2. And there are two data sets, the Swiss cross-validation sets, which consists of many different species, more than 100 different species, and the HPA test set, only human proteins. And you can see that generally we improved the prediction in any kind of recall threshold. And even in the human-only test sets, our space embeddings a little bit better than the Pro-defined embeddings, but at the same time, we are so much better than the DeepLog2. And also, we did a UMAP visualization before we train the model. And based on the UMAP, our embeddings really captured the localization of each protein. And then we checked what's happening in each separate localization. And in most of the localizations, our aligning embeddings it's a little bit worse. But if you concatenate the aligning embeddings with the sequence embeddings, you can always get better prediction results. But especially in lysosome, vacuum, Golgi apparatus, and peroxisome, even our aligning embeddings are so much better than the Pro-defined sequence embeddings. And the reason is that in these localizations, there is no very clear peptide or signal peptide for proteins to get into the localization or get out from there. And it's a very complex of the pathway information encoded in those localizations. And finally, we did a protein function prediction. Here are three aspects: cellular component, biological process, and molecular function. And the cellular component is another way of doing the subcellular localizations, but you got more geoterms to predict. And as I showed before, we can always improve the protein subcellular localization because we can give another complementary information from the network. And especially for biological process prediction, because it's actually predicting the protein pathways. The sequence information do not capture or it's hard to capture the protein pathways. So when we are comparing the aligning embeddings with the Pro-defined embeddings, it's already better than the sequence information. But if you concatenate them together, you can also get better predictions. And finally, in molecular function prediction, because the protein function is encoded in the protein sequence, as defined by the protein, motif, or domains. So in this case, we cannot give other complementary information from our protein networks. And now we have a paper of this project, and we also put all the network embeddings, the cross-species network embeddings, and the sequence embeddings online. So you can just go to the string database and download it. And we also have a GitHub repo of this project, so you can know how to use the embeddings and how we generate the embeddings. And with that, I would like to thank my ex-supervisor, Lars Jensen, and also all the members at Jensen Group, and our collaborators at SIB, Dami and Christian. And we also have very nice collaborators from DDS, and Lisa, Julia, Clara, Daniel, and Rasmus. And most importantly, this project is supported by the supercomputer from Finland. I really love it. And finally, I'm working at Simon Rasmus Group right now. We are doing the genomics, proteomics, clinical health data, and electronic health records. So we are called the multimodal bioinformatics group. So that's it. Thanks for your attention. I would like to hear some questions.
Yeah, thanks a lot, Dewei. I mean,
Thank you.
it's really nice to know that the Finland is also part of this project. I didn't
Yes.
expect that. But why? I mean, just a short question, right? Why is it like that? Because your Denmark doesn't have this infrastructure for doing that, or it's because of some collaboration issue?
I think that they give some like free educations to our university. I think
Okay.
our university is part of like the DEC organization, and I think Lumi is also part of that. So we can
Okay.
get some free education in the very beginning of the development of Lumi, but now we have to apply for it.
I see.
Yeah.
Okay, sounds great. Okay, the way it's really give a nice talk to introducing how the string embedding works and how it has improved the compared to other methods. I
Mhm.
hope we have some questions. So if you have any questions, just feel free to open your mic and ask the questions. In addition, you could also type your questions in the chat. So I may probably start with the very first question, just in case the others are waiting and preparing to ask. So one technical questions I have for your random walk thing, right? At the very beginning, you said that you have used this random walk to kind of align the proteins according
Yes.
to their you know distance or whatever thing, or according to their node interactions. So, however, we know that the random walk will introduce some stochasticity, right? So
Yes.
how is this considered when you use random walk? Did you repeat this for many times to ensure you will converge to a certain reliable data, or how does you do with that?
Yes. Yes. So actually, there are a lot of the methods that you can use to generate the embeddings from the networks without any let's say the node types or the link types. And according to a lot of the benchmarks, Node2Vec performed the best out of those methods. So that's why we choose it. And we also we tried a lot of different hyperparameters. We did hyperparameter tuning using link prediction. So for example, once you have a network, you when you do the Node2Vec, you have the hyperparameters controlling the direction of the random walk. And you can either go forward or go backward.
Mhm.
And we also, in string, we have the confidence score of each link.
Yes.
So you can also use this link when you are doing the Node2Vec. So generally speaking, you will walk to the nodes with higher, or take the links with higher score.
Okay.
But we use the hyperparameters to control the direction, whether it's stay in the same cluster or take longer walk to other clusters.
Okay.
Yeah. And finally, our link prediction is trying to make sure that the node-to-vec embeddings can replicate or to regenerate the original network structure.
Mhm.
Yeah.
So another question from my side before I started to host the other people's questions. So could you also tell us about the training? So how long does the training take, and how did you set the initial hyperparameter values? So
Mhm.
how does this process work? Because you
Yes.
know sometimes it's taking a lot of time and efforts
Mhm.
to do that. Maybe you can share some of your experience on this perspective. Yeah.
Yes. So I will go to this slide. So there are actually two steps or three steps. So for node2vec, you can get the embeddings independently. So with Lumi, I can get at most 100 nodes at the same time. And for the network of like human, it takes like 20 minutes to run. But we have a lot of the fungus species. It takes just several minutes. So when you have such a powerful supercomputer, it's not a big question.
I see.
Yeah. And when you do the link prediction, when you do the hyperparameter searching, it's also fine. Like it just take me a couple of days to finish it on Lumi.
Okay.
And but the most annoying thing is that when you do the hyperparameter searching for the alignment part in the first part, so you have to use the embeddings of 48 species, 48 seed species, and a single training takes like 10 hours. But again, I mean, because I have Lumi, I just make it very fast.
So could you be specific how many resources you got from Lumi for training your model and you
Uh, Let
requested
me
from it?
see. I think I got thousands of GPU hours.
Thousands of GPU. Okay,
Yes. And but for Node2Vec, you don't have to use GPU. Just use CPU and it's very fast.
fine. Well,
Yeah.
sounds very, very good. I mean, at least they are very generous to provide you these resources.
Yeah.
Uh,
And I think without Lumi, I cannot finish it.
Yeah, I can imagine this is a huge, right? I mean, thousands of GPU hours and cool. Okay, so probably some others have also questions. The first question comes from Marcus.
Mhm.
When you are showing the quality control, could you
Mhm.
explain what are the X and Y axes? Actually, I also want to ask
Ah,
this. What
yes.
does it really mean? Because these are probably something we don't know.
Yes.
Hmm?
So this is a cumulative ROC curve, not the standard ROC curve, because when we're doing the pairwise pathway benchmarking or quality control, and we just consider if two proteins they share any pathways, they are the positives. If they don't share any pathways, they are negatives. And in this case, and for some very big pathways, there are like hundreds of proteins. And theoretically, you can connect every proteins in the same pathway. So you have maybe thousands or 100,000 of pairs, but we only care about the pairs with the highest quality. So we use a cumulative way of the ROC curve. So the x-axis is a cumulative false positive, and the y-axis is a cumulative true positives.
Okay, so these are exactly the number of the false positive and the false negatives.
Yes.
Okay. Okay, that's why it's something. Okay. I will wonder why this is actual numbers.
Yeah.
Okay. Obviously, Marcus, thank you for answering his question.
Thanks.
So, do we have other questions from the audience? Just raise your hand or open your mic if you want. You can do that spontaneously. I can shift this microphone to you. But of course, right now we have this good alignment, right? That you show that they are better than other embedding methods. Do you have any ongoing projects making use of this alignment for some analysis, in biomedicine or biology? Maybe you can briefly mention it.
Yeah, I will. I don't have any collaboration, but as far as I know, I talked to some people, or some people also asked me questions on GitHub. Somebody said they are doing like to simulate environment of plants and some of the animals. So they are
Okay.
using the embeddings of the plants and animals. But I don't know the detail of that project.
Yeah, but
But
how does
also,
it
yes.
to use that? I mean, what's this? So the animals, I mean, of course, they can identify the let's say proteins that are close to each other in terms of the network yeah, information as well as sequence information, right? So
Mm-hmm.
are they going to do some whatever well, I don't know what exactly they are aiming for. Of
Mm-hmm.
course, they can do some conservation things, right? To identify those close proteins that probably were not disclosed by just using sequencing information, right?
Yes.
But of course, another thing I would also like to ask, um, have you tried to compare some proteins or very close proteins and using your alignment method to their structure, similarity? Are these really reflecting what it is in reality?
Mm-hmm.
Or
Well,
structural
I
perspective?
I I think the structure information is mostly encoded by the sequence,
Yes.
and our embeddings are mostly giving the network information. But sometimes, if proteins they have physical interactions, they might have also share some pathway information. I don't know, but I once talked to a person that they are using our embeddings to do the homology search in protein
Mhm.
structure.
Mhm.
And I think he's from Finland. He's from Helsinki.
I see. Oh, and maybe he's in the audience. I don't know.
Yeah.
Okay. So I don't have further questions. And how about the audience? Do we have further questions? Looks like no. So now I will hand over to Yulia